Sairam. I offer my humble pronoun to Bhagwan. Good afternoon, everyone, and welcome to the final session of the two-day webinar on physics for healthcare. In the previous three sessions, we have heard scientists, researchers, entrepreneurs from different parts of the globe. It's my pleasant duty now to introduce this guest speaker of this session, Professor and Dr. Håkan Engqvist, who is one of the greatest innovators in the field of applied material science and healthcare. Dr. Håkan is a professor in material science and the director of the Armstrong Materials Academy at Uppsala University in Sweden. He received his PhD from Uppsala University in 2000 in the area of material science. He worked in the industry for 9 years developing dental materials in a venture-funded business. He spent 2 years at the innovation unit at Uppsala University and started a research group. His research interests include the synthesis, microstructure, and properties of nanobioceramic material for hard tissue repair, bone repair, skull repair, and novel methods for drug delivery. He has more than 6,600 citations, H-index of 45, and more than 50 patents to his credit. He has been involved in starting several startups and companies, and he has developed the world's toughest glass ceramic for artificial teeth and is also testing a biodegradable glue that can be used to mend fractures. His company OS Design develops implants made of ceramic materials to repair skull fractures, for example. Silox, which is another of his companies, is a spin-off from Biomaterials, a Venn Excellence Center in Gothenburg, where he was formerly a researcher. Silox focuses on a variety of oral care applications, including toothpaste and gel to treat hypersensitive gums. A more recent startup, Amplicure, works to safeguard medicinal drug against misuse. Dr. Håkan has received many prizes and awards, including the prestigious Innovation Award from Uppsala University for successful knowledge transfer. He has been working on various innovations in the last decade. In one of his interviews, he says, I quote, I am a fairly strong believer in integrating the knowledge triangle. That's how I work. My knowledge triangle comprises research, education, and innovation, but may also contain other forms of collaboration. I end quote. Despite leading many companies, Professor Håkan considers himself still a researcher. Please join me in welcoming the material innovator, Professor Dr. Håkan Engqvist. Without delaying further, let's listen to some of his great innovations. Professor Håkan, we request you to take over.
Thank you. Thank you very much for the kind introduction. I hope that you see my screen now. So I will go through, present some about the university and then some about how we work and go through some specific projects. And then in the end, I'm open for questions or so on that you might have. So I come from Uppsala, where I work at Uppsala University. And Uppsala University is, it's relatively far up north in Europe. And we are the religious center, or used to be the religious center of Sweden. And we have a dome that was relatively old. And at 1477, we got the permission from the Pope to start to teach. So the university, and this is the same as many universities in Europe, it's connected initially towards the church that the Pope gave permission to teach, and from that later on it developed into the university. to a full-fledged university. So this is an overview picture of the old part of Uppsala. It's very close to where I live. And the Ångström laboratory, where I work, is farther away in another campus area than what you see in this picture. Okay, so Uppsala University. We are quite a traditional university. It's called the Humboldt. The way it's run is called the Humboldt University. That's the same as let all flowers bloom. So there are many research groups, and we are relatively free in a way. It's a lot of tradition connected to the students. We have roughly 50,000 students in give and take some, depending on how you calculate. We are highly ranked. We are normally top 100, maybe 80 in the world, according to many of these different ranking systems that are used. Some profiles, Linné, ordering of the plants. Celsius, the thermometer comes from here. We have found quite many of the elements in the periodic table. For example, yttrium. It's a small place outside Uppsala called Ytterby in Swedish. That's where they found that element. But there are other elements that have also been categorized and found here in Uppsala. We were also the first to splitting the light, the light within the wavelength. Okay, and that, yes. And we also discovered the lymphatic system. So there are many things. We were quite prestigious, and we've done quite a few of it, especially in materials, chemistry, and medicine, I would say. Nowadays, we are very strong in what's called peace and conflict research. I would say maybe that and evolutionary biology at the moment are the two areas where we have a really, really strong position. Some Nobel Laureates that we have had at the university, two of them, one of them I know that I worked close to is Kai Siegban. He got the Nobel Prize in Physics in '81 for the ESCA or XPS. I used to sit in the same corridor as him. Okay, and I then work at the Division of Applied Material Science, which I run. It's my research division. And we work at Ångström Laboratory in Uppsala, which is a relatively newly built materials or science. Nowadays also would be computer science and mathematics, but it used to be just materials. Research center in Uppsala, and it's relatively well equipped, I would say. We have access to most of the equipments that you could use for this type of research. So, as mentioned previously here in the introduction, so my personal background comes from what's called, in Europe, this is very modern, it's called the knowledge triangle, or K3, where normally researchers and as we that are participating in this workshop conference, we are located on the crossroad between innovation and education. Okay, that's where university are, and Uppsala University is the same. Some people more towards are more towards creating how should I say research. Others are more towards educational part. But in reality, we are also then connected to, in this case, what I call innovation, but that could also be collaboration with society. And if you do this as a triangle, in this case, you can point yourself to where do you belong in this triangle as a person. Not only on creating knowledge and spreading the knowledge, but also how to collaborate and spread the knowledge out to the society. So it's a triangle. My personal view, I do not teach that much, so I'm much stronger between the creating knowledge and spreading that through innovations. So more leaning towards that side of the knowledge triangle. And why is this important? Because it creates faster societal impacts of research results. For example, in our case with healthcare, results that reach the clinic gives a higher impact. I'm 100% sure of that. Especially long term for universities, this is very important. If we work with healthcare, of course, if what we do could reach the clinic or societal use, the impact goes up. For the students, they are trained for future jobs, plus creates a good network if they are more broader in sense. And knowledge triangle normally means that you need to collaborate to reach critical mass. And it's very, very difficult to take advantage of excellent research without the integration of knowledge triangle of what you do. So this is what I'm a strong believer in. And it's basically to turn money into knowledge. That's what we do. We get tax money, we create knowledge. Next step is to create knowledge into money or societal needs. That could be, for example, create new laws. That's also a way to spread the money that we got from taxpayers back to the society. Or we could collaborate with the existing industry, or we can form new companies. For example, there are many ways of doing this. But you see what I'm after, that we get paid to do something, we create, and then I find it's also our job to help to transfer that what we have created back to the society. Okay. So more specifically, what we do in my research group, which is called Materials in Medicine, we do spare parts, regenerative medicine, mainly for hard tissue. We would like to move more into soft tissue, but mainly hard tissue. That means the dental part. CMF is the cranial, spine, joints. Yes, and we do drug delivery and some sensors, not that much. I will go through a few of these examples. And I would also say the main part, what I do as my special part is hard materials, ceramics and metals. We have been mainly working with ceramics. We are spreading into doing our own metal as well, metal alloys. In Sweden, metal and metal alloys are very, very strong. We are a metals country, and the industry is strong, and there's a very strong institute sector. So I've been thinking, I shouldn't go there, because it becomes too complicated to run projects when there is so high competition in the country. But recently we have had quite good ideas on new alloys that we are exploring, and that is both connected to new needs plus the new possibility with the additive manufacturing. So we are working more towards metals. I will not present that today at all, but we are doing that. So what we normally consider, what and how can ceramics help human health? How to design and prepare better ceramic-based materials. So we do structural ceramics. That means that we go for strength, optical, or stability. And normally this is a composition structure, grain size, type of loop that we go through. We work with functional ceramics for bone, teeth, drug delivery, and also antibacterial. The applications are normally clinically driven, and we are always interested in better and more ceramic materials. We try to do more and better with less. So we made a small image here of what type of materials we are working with, and also describing in the let's say, the part of the periodic table that we are working with. You can see there are quite many of these of the elements, but these are the ones that we try to combine into new materials. So what do we synthesize? We do structural ceramics for load bearing. Mainly these are silicon nitride based. We spent almost 8 years on this. Quite interesting area with silicon nitride. The background is that there was one innovative company in the US who's been doing silicon nitride for spine that we have been following quite a long time. Not that much research done, but from a company perspective, they have been doing quite well, They have accumulated a lot of venture capital. So I've been following them, and then we tried also to follow their example, and we also been working with silicon nitride. We are relatively known about this, but it's a very interesting material. It's very high mechanical strength. It could be biodegradable. It is antibacterial. It is bioactive. And so we are doing this for load-bearing applications. That means in the spine and in the joints, especially hip joints, but most likely, and I'm also interested to go for the small joints. We have developed up to the far right corner here, we have translucent strong glass ceramics. We have developed the world's strongest translucent transparent material that we are pursuing in the dental field. Okay, then nanostructured ceramics, we do, that was also mentioned in the short bio here, is we have developed a method to do small ceramic particles that we have now, is actually now being sold in tooth gel in the US. So we use it for a tooth occlusion. And then a very interesting area is to see the morphology control of calcium phosphate. How could we control the growth and structure of calcium phosphates? We have been working with that for many years. And then in the further down to the right, we have additive manufacturing of ceramics and gloss ceramics. We are trying to do, since we come from the material side, we don't from the design, we're trying to mimic different types of designs with our materials and additive manufacturing. Some applications I mentioned, spinal fusion is one, with silicon nitride. We have a lot of work done on dental, both because I worked almost 10 years in industry with developing dental materials, and then I continue at university. We do restorations, anti-hypersensitivity. We have been doing a lot on whitening. We have been doing on tooth fillings, and we spent a lot of time, actually most of the time, on dental implants. That's the titanium part that is kind of replacing the tooth root. And then functional bone grafts, I will talk more about that later today, and that is bone graft injectable materials with specific functions that could be injected directly into the bone area. And this is how it's normally done. Could be done then, it's instead of having open surgery through materials, the surgery could be done injectable instead, which saves time, cost, and pain for patients and for the hospital and for society. We have been doing a lot about this. One thing that I would like to present more about is this tissue adhesive that we are at the moment very intrigued about. And it has to do with, at the moment, there are no bone adhesives that could be used clinically. The polymers that are used in the clinic are non-resorbable, and they don't really adhere to tissue. There are a few, but they are relatively weak. The one is called Tisel. It's a fibrin type of glue that is used to glue skin and stuff like that together, but that doesn't have enough mechanical strength to glue pieces of bone. So instead, if there is, for example, in this case, an elbow fracture, that's what you see in the middle picture there. So many fragments, it's ridiculous. So they are all placed in place again with wires. So that takes a long time in surgery. And it's very hard to do this clinically. So to have an elbow fracture is complicated to fix. And this is just one example where glue would make a lot of sense. There are many things also in the foot that are complicated to fix. And fingers, there are many areas where it could be very beneficial. We can glue the patient instead of having all of these wires. So we've been working on a resorbable bone adhesive. And what we did, the first experiments were done 2013, where we glued 2 pieces of bone from the slaughterhouse. It's just animal bone that we glue together. And then just to measure the strength after 24 hours, we put buckets of water in a type of homemade mechanical tester. And we were able to do 36 kilos before it broke, before the fracture broke. So then we started to realize, actually, this is something really, really strong. And this is from a cadaver workshop where we were able to glue pieces of bone together in 2017, 4 years later. And we are now targeting to do wrist fractures, in this case, together with the screw, to glue the screw in place. So this is a regulatory question that we can talk forever about. But that's the way we're pursuing this now, and we are working really heavily on this. And I would say that if you just to imagine how many steps, so if there is a wrist fracture in a patient, these are all the steps that are needed before that patient is discharged. And then you have the payment journey. And if it doesn't heal, it goes back again. So the cost for this is around, the cost of this is around 60,000 Swedish kronor or 6,000 euros. I don't know the conversion rate to India really, but that's roughly what it would cost in Europe. And with a bone adhesive, the cost would go down to around 2,600 instead of 6,000 euros. So there is a huge saving for society and for patients, of course, if they're glued together. So why haven't that been done? And if you go and search in the literature scientifically, there are many, many people who have tried to do bone glues, many. Many come from the muscle adhesive. And some are just polymer based, but quite many, come from the muscles that attach to the boats. In the bottom of the boats, you have all these muscles that could sit on big ships. So they've been studying a lot of studies on how they are actually made, and then you try to replicate that and make it into glue. And most of these have failed in the investment regulatory human clinical trials. That's where they fail. So it's a very complicated project to run towards the clinic. It's easier to do it in the bench, I would say. And so far, no one has made it to the market with a bone glue claim. And we are working what is called the phosphoserine. And if you, for example, have a broken bone and you try to image that and mark for phosphoserine in our bone, you would see a lot of phosphoserine is already present within our own system. So we are then we are using the pure phosphoserine and added that to our material systems to get an immediate glue effect towards tissues. I normally say that what we do is an injectable high surface energy cement. That's what we do. And it seems to work. They are all degradable. So far, we have been doing rodent study, and we're going into bigger animals. So we mix it. We try 3 calcium phosphate and phosphoserine, and we're able to even measure these underwater. I don't need to go through all of this, but we made a lot of shear tests and pull-out tests on these. That's how it could look like in powder, and after hardening, that's how it normally looks. It looks more or less like a glass surface. These are just shear strength values as a function of amount of mole percent of phosphoserine. So there is an optimal between, let's say, 20 and 40% of phosphoserine in our formulation. There are many formulations that could be done like this, and if you're interested, you will also see the phosphoserine is just one part of a very big family of chemistry. The compressive strength is very, very high, and this I don't need to go through. We are testing the safety efficacy in vivo. We have now published in both ex vivo, but we also now have our first in vivo data coming out from rodents. Very complicated, I would say. very complete way of doing, because we need to develop the preclinical models doesn't exist. So we need to do them first before doing it. And I'm a material scientist, so it's quite complicated, and it's a lot of work being done on this. Very interesting project. So these are just some initial data from a femoral bone core model, and so this project is progressing. It's now we are running it now in Uppsala. It's in Gothenburg. We have recently started to collaborate with the veterinary hospital in Uppsala, the veterinary university in Uppsala. It's in Gothenburg, and we have a lot of activities in Ireland on this with the bone adhesive. So if you have questions, you can take them after this presentation if you have any question on the adhesive. Now I move on to next area where we have spent a lot of time is the cranioplasty. And cranioplasty has a definition. It's the restoration of a defect in the cranial bone or correction of a deformity of the bone that may happen after trauma, as in a depressed fracture. Okay? And they're looking at old skulls, there have been in excavations and so on, there has been shown, of course, that trauma and different diseases has been common to the skull bone. And it's quite complicated to fix. So what could be done? So if you have a big defect in the skull bone, in the interim, normally you use a helmet. You need to protect the brain. Bone then could be boiled and replaced. They could be put in alcohol and then autoclaved and replaced back. So then you use your own preservation of autograft. And then people have taken the bone flap in the abdominal wall, very traditional way, complicated as you could imagine, but it could actually work, but that is normally too expensive for society to pay for. Then you can take the bone flap and put it in a sterile, may have it sterile in a -70 freezer for many months until everything within the skull bone, within the brain is fixed, and then you can replace it. And then you can just autoclave it or do ETO sterilization. Today, I would say most hospitals are using the bone flap -70 freezer model. They resorb over time. So when you replace your own bone, it normally resorbs. If the defect is too big, the old bone resorbs and goes away. Okay, so then you have a hole in your head, and this is the clinical need. So what has been used to mainly titanium and PMMA. That's the two materials that are used clinically. And the problem is the soft tissue. So what is needed is to have a method or a new material or a new design that could help to solve this problem. The number of patients are not that big. I have no idea how many it could be in India. Actually, I have an idea how many it could be in India. And compared to the total population, of course, it's rare. But for the actual patient, it's horrible, I would say. And if they have this problem that you first have had a disease, you lose your skull bone, you get an implant, and then you have a problem with that implant, you're in a bad shape. And the cost for society for each of these patients is in the order of hundreds of thousands of euro is the societal cost. So if you look at from patient suffering, that's more of an ethical, moral thing. And then it's also a real cost for society. So me and a plastic surgeon, Thomas Engström, we founded the company Ostdesign. And so I've traditionally, this is how it could look like with a titanium mesh. And you see them now in place there. Let's see. Good. So what we did, we developed this one. Yes. So we developed a composite implant where we have a ceramic on top of a titanium mesh. So we use the titanium for load support. And we have with 3D printers, so it's a CAD design. We can talk a long time if you have questions about these methods. It's very, very interesting, and I would say there is a lot of innovations to be done on this computer side, not on materials as well, but mainly on the computer side, software. So we combine these two materials into one implant. And with the design, we do a mosaic design so you can have blood flow and even up the pressure from the skull bone. It doesn't need to be a high pressure. Many of these patients otherwise have a continuous headache due to high pressure from the fluids surrounding the brain. Okay. And this is how it could look like in place. What we have shown now is, I will show you data points on the clinical outcome. We have made incisions in patients. So this is from a patient that he had a gun wound. In this case, I remember this, he had a gun wound. And there were something protruding. So we were able to, the surgeons open up, and then we were able to take small pieces of the implant after implantation. And we did analysis, or a lot of analysis of this, and this was after 4 years, and we can show that we have got new bone formation far away from the bone. Very, very interesting. Where you will have all the components of a living bone. So, okay. So some clinical results, and these are just published data, and this was from our first 100 cranial implants. We have an infection requiring implant removal was below 2%, and the competition showed data everything from 5 in one of the cases up to even 40% in some cases. And now we have passed now over 1,000 implants sold, and the data still are in the order of this 2 to 3%. Okay. Now it moved here. Now, so you can ask, now I leave the cranioplasty and I go into bioactive materials, which is an area where I've been working a lot. And biomaterials were first generation, were designed to be bioinert and not interfere with host tissue, like PMMA. Second generation were designed to form an interfacial bond with host tissue, would be the bioactive. And the third generation biomaterials are designed to enhance biological effects, to remineralize, or to be more biomimetic. And we have been spending a lot of time on bioactive and biomineralization materials. And there is an ISO standard even that one could follow. And I think there is an ASTM file standard as well on how to test and evaluate these materials. Normally, it's a soaking method. It's a very low-cost method. You do your own simulated body fluids. You soak the materials 4 weeks, 37 degrees, and then you do surface analysis, various methods, and then you study what has been formed on the material. In our case, it's normally we look for hydroxyapatite. The mode of action is, I would say, negative surface charge. That could be bioglasses for those who work with glasses, quite an interesting area. Calcium phosphates, and then there are some other material systems that could be used. And I encourage people to read the papers mainly from Kokubo, Japanese, very well-known researcher within the field of bioceramics. He has been doing most of these mode of action studies. I will go rapidly through these, and what we are after then is a dental material. In our case, now a dental material is that towards the host tissue form not only a micromechanical bond but a real chemical bond. So to summarize, the advantages of this is bioactive nanobiomaterials offer several advantages. It's bioactive, they have the potential to remineralize the host tissue, in this case is teeth, and the potential to hinder growth of bacteria. One last topic that I would like to talk some about is it's an area where I think maybe this is one of the 2-3 areas where I spent most of my research time is on this artificial tooth. So I mentioned the crown with the glass ceramics that we're working on, the high strength ones, high strength translucent materials. And then when a tooth is replaced, the root is replaced by a titanium screw. And reading literature, titanium has a quite good clinical outcome. But in reality, this is starting to become more and more questioned. Sometimes it's a functional replacement of the tooth, and sometimes it's aesthetics. So, but to form a stable interface between bone and an artificial material like, in this case, titanium, it is complicated. And on top of that, it's protruding into the oral cavity. So it's always a pressure for bacteria to enter where at the gum material interface. And if that happens, I will show you a picture later on. What that means is that the bone around the titanium implant resorbs, goes away, and instead leaves like a void. And in that void, there are normally plaque formation and bacteria biofilms, and it leads to and on top of that, these patients, when you have a tooth replaced like this, there are no nerves there, so you don't feel anything. So the occurrence of what's called periimplantitis is difficult to detect. So dentists all over the world are debating what is periimplantitis, how can it be detected, the occurrence of it, and how then to treat. It's a huge opportunity research-wise, but it's also a complicated area, and I like to be I like to be that because it's a lot of things being done, and it's a high interest area, I would say. So metal implants are skin penetrating in this case, what we've been working on, and skin penetration means a risk to future problems. So they have to form a stable unit with the surrounding tissue. That means both bone and soft tissue. There are sometimes stability problems due to poor bonding between the metal and the bone, and then you have severe long-term problems. And then there is also the lack of local blood circulation, and it's difficult to treat. And this is how it could normally look like. This is what's called a flap surgery around an implant. You can see the void between the teeth, and you see the artificial tooth on top of it, and then you see the void that has been formed due to periimplantitis. And this void could either be fixed later on, or it needs to be avoided to start from the beginning. And we have projects working on both of these. And we do the first one, maybe that could be open for collaboration in this setting. It is for surface modification. And it's to make the implants bioactive, and also, which is more complicated than antibacterial. So we do activities on materials for bone integration and regeneration. And we do that also by incorporating of active trace elements into the coating and surface modification. I'm not going to really go into details due to maybe proprietary reasons on exactly what we're doing at the moment. But we are working with negatively surface charged of titanium, and we are doing that to enhance the bone integration and reduction of infections. And we are also having projects running on when there is a void formed, how to fill this void with new material solutions. So we do also have projects on that, which is very, very interesting. Okay, so I think that was about what I planned to talk about. I think I had roughly 45 minutes to talk, and so any questions or so, I'm yes, all yours.
So I have one question.
Sure.
In your tissue adhesive material, when you're joining the two pieces of bone, there is definitely a probability of susceptibility to infection internally.
There is.
So how do you check that? because it can be a center for extra bioactivity and extra cell growth. It's after all a substrate, new substrate that is added.
Yes, it is. You're right in that. So this is one of the, what you bring up is a good question, and it is for us complicated. So how should I? There is a new substrate, and that's that between the two pieces of bone, and we would like it to be mechanically strong for a period of time, and then magically be transformed, resorbed, and this joint, this area where they used to be should be then instead grown together and just be bone instead. So you heal the fracture. And so that's why we're now moving into bigger animals. We've been doing the small animal work, and we're moving into the bigger animals to get a more long-term perspective and bigger amounts. The complicated part to test your question in the small animals is the amount of material that is used in these small rodent bones. I don't know how, we have very good data, but I don't know how representative they are for the human size. So that's why we're now moving into sheep studies, where the size of the fractures will be bigger. So we can test that question more in detail, because if it stays too long, if our material stays too long, I think the effort that we are doing will not be successful. On the other hand, no one can really say what should be the resorption time. Should it be a week, 3 months? I suppose a year is too long. So we don't know that. We need to test.
Okay.
Yeah, no one knows the answer to that question, the real answer. That's why we need to go to big animals and test.
So just one more question.
Sure.
When you are given a different type of material in your table you mentioned, and your material with phosphoserine, the infection level, definitely what you have shown has come down. I just wanted to know, can we not, now that we have gamut of so-called nanomaterials which can to some certain extent control infection, have you tried using some of them to reduce the infection almost below?
No, not really. It's a regulatory, very complicated question to add. So we are working with well-known components, and some of the nanomaterials that are publication-wise said to be antibacterial or reduce infections. One should also know that some of these infections that arise, especially in these super big defects in the skull bone, they could have been there. Many of those are there already from the beginning. So the reason why they replace the bone is that there is an infection. So the bone is, and then just to remove that surgically, that bacteria or that infection is complicated in the operating room, and it has nothing really to do with the material. So it's already there when it's placed, and then it grows. So then comes your question, should it be something else added? So what the surgeons do, they soak it in antibiotics.
Okay.
Some of them. Some of them do that. That is nothing that the company is promoting or so on, but we do know that some do that. And then to add other components to the materials that could be antibacterial, I would say to lower the infection rate below 1%, I would say the cost and the benefit of that is low. That the cost is too high. I don't think anyone will do it. That it needs to be some other clinical application or so on, because with even bigger problems, I would say.
Okay, thank you.
Yeah.
Professor Håkan.
Yes.
In this bone glue, bone glue that you mentioned, the phosphorin is a unique one, or are there any alternative to phosphorin?
Phosphoserine is a family of, there are many analogs to phosphoserine that could be used. We have been working with phosphoserine by historic logic reasons. There is a long story behind why we started with phosphoserine, but we have published, I don't have that in front of me now, but we have published papers where we describe other. So the serines there are many serines. If you search for this online, you will see there's the serines is a family of molecules. We work with the phosphoserine, but there are others like the creatine, for example, creatine. There is, if you search for it, you will find, I think they should be published now online. There is a family, and some of these are, and at the moment, some of them could be better than phosphoserine, especially referring to the previous question about the risk of the resorption contra adhesion. It could be so, okay, now we are working with phosphoserine. And it could turn out within a year or so, 2 years, I sit there and I know that, okay, phosphoserine didn't work because it resorbed too slow. We should have chosen another one of these analogs and gone with that instead. It's a money thing also. We cannot afford to run big animal studies with many of these analogs. So we have chosen phosphoserine, but it might be wrong because there are others that could be tested that have about the same effect in mechanics.
One more question. You mentioned silicon nitride based implants, dental implants, but silicon nitride, if we see color wise, it will be black, right? But though it is hardest material and used in hard coatings, so is it a good one to use for bio implants, aesthetic reasons?
These implants are within the body, so no one really cares if they are black or white. Within the spine, no one sees it.
Dental implants I was mentioning.
No, dental implants, I think it's biologically, it's a very good idea. Clinically, I don't think they would use it. By aesthetic reasons, they would go with others because they are gray in color, so they wouldn't use it.
Okay, so one more question. This antibacterial nature of this bio implant. So what elements you are preferring for these bio implants? Bone
it depends. It's different for different. With the silicon nitride, it's just the resorption of silicon nitride releases ammonia, and ammonia kills most of the bacteria.
Okay, thank you.
Hello, Professor Håkan. There are a few more questions on the Google Meet chat. So by the students. So I'll just ask one by one. The first question is, how much time does the glue take to harden?
Minutes.
Minutes.
A few minutes.
Okay. The next question is, are zirconia implants better than titanium ones?
Yes, in a way they are. The drawback is the fracture toughness, because titanium is more secure from that perspective. But I do believe that silicon nitride have biological benefits compared to titanium. That I would say, and that mainly have to do with the bioactivity and antibacterial properties of silicon nitride.
Okay. There's another question which is about nanomaterials. It says, do nanomaterials create any health problems when they are used in the body? Toxicology.
Sure. So this is a big topic. We always keep away from non-resorbable nano-sized particles, because there are too many issues to be answered with those. So, and we are not toxicology experts, so most of the things that we work with are resorbable and go away. And then the toxicity is not an issue. But I do believe if there is small particles of non-resorbable nature, it's complicated, and we don't work with that.
Okay. One more question is,
sure.
How much time your study on this biocompatibility should be done before you can arrive at at a safe time?
You mean for to go to clinic or animals?
Yes, clinic, sir, clinic.
Oh, there is in Sweden or in Europe, there is this ISO standard 13485, and normally we just follow that standard. And in the standard, they pose what type of studies that needs to be done. So there is how should I say, communication with the notified body, and we go through, this is the project we have, and what type of studies is recommended that we perform. In some cases, it could be very little, and in other cases, it's a lot. Like with the bone glue, that's a lot. It will take, before that is in the clinic, it will take many years.
Thank you, sir. So there's one more question on my personal behalf. When you fill something in the bone, we know that as we age, the bone shrinks. So have you done any long-term studies to see, like, because the other material, will it also degenerate at the same rate, or does it create any non-uniformity or something like that?
Not really. The materials that we work, they all resorb, and they would resorb. We try now to make them resorbable in a rate that could accumulate the change in the bone, so they wouldn't be, leaving a void or stay too long. I would say calcium phosphate research have come to that level of knowledge at the moment that we know, okay, for this specific application, we should use this composition. So we create a composition that would work. And that's so no, I understand your question, but it's not really a big issue that we've seen at the moment.
Okay, sir. Since we have 3 campuses participating in this, I would just like to ask if there are any questions from Anantapur campus.
No, ma'am. No questions.
Okay. Thank you, sir. Thank you, sir, for your wonderful presentation, for taking out time to be with us. We really enjoyed your presentation here.
Thank you.
Yeah. And I hope many have got inspired to take up research in this field, which is a very, very important field in medicine.
I hope so.
We also welcome you here, maybe at a time when things are better, and we'll all sit down together and listen to you.
Yeah, sure. I will come. I will talk to Sanjeevi and make arrangements. Yes.
Yes. Thank you. Thank you very much, sir.
Thank you very much. Thank you.